Great. Thank you. Well, thank you to the organizers for giving me this opportunity to speak to you all. I really do appreciate it, and also the organization that has gone into this. Yes, I'm going to talk a little bit about sample environments, and the reason for that, I guess this is going to look a bit like self-promotion, I guess. So my colleague Elliot Gilbert, who's the SANS instrument scientist for Quokka, we were asked to write this review for advances in colloid and interface science. And it was on advances for sample environment for neutron scattering. And when we started this, we thought it was going to be something that was going to be quite short, but it didn't actually end up being that. It's quite a comprehensive review. And this is a picture of the graphical abstract, which is a bit blasphemous because it's based on the SANS aspects, and this is a reflectometry meeting. So what I'm going to do in this talk is go into a little bit of sort of like the literature analysis we did, and then just some examples of stuff that we've done at ANSTO and possibilities for improvement. So we did some literature surveys, and I don't know if anyone is familiar with this software called VOS Viewer, which is a way of, if you do a literature search, you can then sort of determine sort of the relatedness of the key word terms of all the articles that are in the literature search. And we did this for using the search term that you can see on the screen. So it's like neutron and sample environment and scattering and tomography and imaging and reflectometry. And it gives you what you can see on the screen, which is this network of the related terms. So the round balls are the various different terms, and the bigger the blob, the more times that keyword appeared in the search. And then the lines are sort of like how they're connected to one another, and the different colors are clusters, which are sort of like a cluster means sort of like those terms are sort of like closely related to each other. And the things to point out with this is sort of at the green cluster at the bottom is sort of, I guess, sort of like most of the terms you would expect to find with sort of like hard matter studies and magnetism, whereas for the purposes of what we were doing, we were interested in sort of like the more soft matter aspects. But what we found is sort of the terms that you would associate with soft matter was sort of spread across all the other clusters, so across all the different colors. And what we interpreted from that, and it's certainly very much open to interpretation, is sort of we found that sort of like for soft matter applications, a lot more sort of bespoke and specialized sample environments tend to be used for experiments. So this diagram is sort of for neutron scattering in general, and I tried to make that specifically for reflectometry only, and that only resulted in 34 hits, which unfortunately is not enough to do this type of analysis in VOS Viewer. So I tried to do sort of like my own grouping, which is very much my interpretation, and sort of like of that 34 papers, only sort of 7 of the papers were for specifically on sample environments, specifically for reflectometry. So not very many. However, so like 9 of the papers were sort of, I guess, sort of reflectometry papers, and for some of them, if you looked a bit deeper, although the paper was principally about the scientific question that was trying to be answered, they did actually have to create some sort of bespoke or special piece of sample environment to get the experiments done, even if the paper wasn't necessarily about that sample environment per se. So yes, so maybe one of the things to think about is perhaps when we have a new piece of sample environment, we could perhaps publish it more. So to go into sort of like a few things that we've been doing in ANSTO that I think have been quite interesting is one of the things since we took on SPATS and installed that, and its vertical sample geometry has meant that we've been able to install sort of like the ILL type sort of blue niobium shield high temperature vacuum furnaces, and that's sort of like really come from some quite cool experiments, and sort of like my colleague David Corti has sort of been really instrumental in finding some really good science involved with this. And one of the things we had to do was make a holder to go onto the end of the stick to hold thin film samples. So we have this stainless steel holder, and you can just clamp the sample into this. We have actually modified it a little bit because if you go to really high temperatures, you do seize the bolts, which makes getting the sample out a little bit difficult, especially if you want to keep the sample post-experiment. So now we just use tantalum wires to hold it to this sample. But some of the experiments we want to do, which have been in conjunction with the University of Technology in Sydney, with Ashware, who's a postdoc, and her supervisor Francesca, is they're interested in looking at forming epitaxial graphene layers from silicon carbide, and they do this. They have this process, which you can see in the graphical abstract. So they take a silicon substrate, deposit silicon carbide, then deposit nickel, then a copper layer, and then they heat treat that. And during that heat treatment is when the graphene forms, and then they can etch away the nickel and the copper to leave the graphene layers. And what we looked at was the heat treatment stage in situ with the furnace, and the first step was to heat the sample up to 600 degrees C, and in this you lose all the Kissig fringes. Reflectometry becomes quite featureless, and the surface comes rough. But then you heat it further to 1,100 degrees, and then in the SLD profile, you can start to see this carbon layer forming. Although there's still some uncertainty, so if you look at the uncertainty, they're quite large. However, we were unable to fit the data without this carbon layer forming, and the group took the samples and did other XRD and Raman spectroscopy to confirm that the carbon layers have also been formed as well. So that was quite good. We think with this experiment, 1,100 degrees C is the highest temperature experiment ever done for reflectometry, although we're still waiting for someone to say, no, we've gone to higher temperatures, so now's your moment to speak up, or later if you want. In the
I
literature,
will
the
I will
nearest.
sp I will speak up.
Yep, go
So
for it.
that's no,
What's
sorry, I will comment later.
Yeah, no. I mean, when we looked at the literature, the nearest we were able to find was 850 degrees C, but I'm sure there's someone who's done higher. So it'd be interesting to know. Um, some more recent work, Jeff McCallum's group at the University of Melbourne and his post Menjif is they've been interested in forming these vanadium silicide layers, which are superconducting below um, 15 Kelvin, and they have on silicon a thick layer of silicon oxide with vanadium, and then um, they thermally anneal these at 700 degrees C, and essentially they form a vanadium oxide layer and a vanadium silicide layer. And with using event mode, um, uh, we were able to show the progression of the chemical pathway of these films. So um, we use solid liquid cells a lot, ACNS, between our two instruments. They probably account for about 70% of all the experiments we do. So in the photographs here, we've got our setup on platypus and our setup on spats. And we use silicon backing wafers because we quite like the nice low background on them. But the massive downside for using silicon backing is that they are very high maintenance. Silicon breaks easily, which means they have quite a high replacement rate, and we have to get someone to drill the holes for the inlet and outlet tubing for exchanging solutions. So they can be a bit of a bind. So we're trying to redesign our wafers so that we have um, a more robust cell, but still trying to maintain the low background or lower background that we can achieve. So, and there's many different cells here. I'm not highlighting these for any particular reason because everyone has their own design of cells, which has their good features and maybe not so good features. I just chose these two because they were images of solid liquid cells I was able to find. So you've got ones with peaks and magnetic stirrer bars for mixing solutions, etc. Sort of going back to the background, so we've recently tested some different solid liquid cells that um, various people have provided us, and we do find that silicon does give us a much better background than peak and PTFE. But so now we're just trying to weigh up whether we sacrifice some background to have some more robust cells at the moment is what we're trying to grapple with and trying to find a solution of, well, can we have both? Um, Another innovation from Isaac Gresham, who's a research fellow at the University of Sydney, is he's been 3D printing his own solid liquid cells, which you can see here. And this is one of the things that we sort of like highlighted in the review is that 3D printing has really come into its own, and you can certainly find a lot of 3D printed devices. So it's a very useful technology for making things quickly, which is really good. So changing track, we do a lot of atmospheric control, whether that's gases or humidity or vacuum. So some work colleague Ian has done, which I also helped him with, is to look at bulk heterojunctions. These are you have a donor molecule and an acceptor molecule. The donor is this polymer with an impossible name, and then you have an acceptor which is ITIC, and he's studied different blends of them, and under vacuum conditions, heated them up to look at the thermal annealing and how the aggregation of the ITIC affects the performance of these thermally annealed devices. And then we've also had people from the Max Planck Institute come over and use our chamber. And they've coupled our chamber with a four-point probe, which they brought. So we can do further in situ measurements in the chamber, and they were looking at hydrogen exposure to nickel thin film. So you can see it has the presence of hydrogen in the film. The SLD goes down. That was quite a good piece of work, or a very good piece, excellent piece of work, I should say, really. For spectroscopy, so if anyone knows anything about the SPAPS instrument, it used to be the BioRef instrument at the BER reactor at HZB. And one of its unique selling points was it can do simultaneous FTIR spectroscopy with NR. And we have continued to do that. So this is some polystyrene here that I took when we were commissioning, which was spin coated onto silicon. And yes, we're now in the process of thinking of another way of doing this, because unfortunately the IR spectrometer is a little getting a little elderly in its age for spectrometers, and will soon need replacement. And I found this recent paper from Rainer Dernhint and Thomas de Earth's labs, where they've combined IR infrared spectrometry and elipsometry all into one measure or one sample simultaneously. And I think this is pretty cool. And definitely simultaneous measurements are the way to go. Unfortunately, I don't have a photograph of this. I couldn't find any, but with one thing that we've done recently with electrochemistry is we've had a couple of users that have wanted to couple measuring their electrochemistry, but the interesting electrochemistry is at quite high temperatures, like above 100 degrees C, so higher than what we can do with a conventional water bath. So we've developed a high temperature electrochemical cell, which can go up to 200 degrees C. It also, unsurprisingly, doubles quite nicely as a high temperature solid liquid cell, even if you don't want to do the electrochemistry, which we've had a group do as well. And finally, another thing that I think we do quite well at ACNS is the event mode, mainly thanks to Andy. So we can time slice the data post measurement during reduction. However, if we want to couple this with changes in sample environment, it's a little tricky because at the moment we have two data files. So one thing that we would like to do going forward is also sort of having time slice the sample environment data all in one file. So to summarize, reflectometry requires a large variety of sample environments, perhaps may go far as saying larger than other neutron scattering techniques. That's kind of what I found from doing this review. Um, but there are a lot of core sample environments, but also many bespoke sample environments. And maybe one thing we should consider is so for the core sample environments, sort of like some sort of standardization across the facilities. So when we've had users bring, say, their solid liquid cells, which they've used at one facility many, many times, and they bring it to us, we have challenges, because we're sort of like, well, we don't have those sort of connectors, or we don't have those sort of fittings, or we've got to make some sort of adapter. So maybe we could make things a little bit more efficient by sort of like considering some standardization for things like that. So I'll just finish on some acknowledgements. So obviously, a lot of what I've presented is part of the team's work. So thank you to the reflectometry team at ACNS, and also our fantastic sample environment team. They're actually, given what they have to do, they're a very small team, and they really do put a lot of effort in to get things done. It's really quite amazing how they do get things done on the limited resources they have. And also to NCRIS, who part funds the ACNS functions. So with that, hopefully I've stuck to time. Thank you for listening. Are there any questions?